I work in New Zealand, but I'm actually not from New Zealand, which you might pick up with my accent. But I'm happy to speak here. Thanks, Rob, for the invitation. Right after the conference dinner, which can be a bit challenging maybe, but we'll give it a try. So my research has been on human T cells and dendritic cells in the last 10, 12 years. And when Rob asked me to speak, he asked me for a title, and I didn't really know what to present at that point, so I'd give a very broad title. So when I made the presentation, I stuck with it and present a little bit of everything. So when I started my research, first the plasminogen system, like everybody presented. So a few topics that we're going to cover today is T cell activation and the role of the plasminogen system in that. The cleavage of the chemokine CCL21 and how that affects immune cell migration and T cell adhesion to endothelial cells. And then last, I want to share some preliminary data on dendritic cell endocytosis of dead cancer cells. But first, when I started all this research, we had a look at the expression of some of the plasminogen receptors on different human immune cell populations. And we found all of them actually that we looked at expressed on the immune cells that we looked at, which were monocytes, B cells, CD4 and CD8 T cells, and natural killer cells. There was a bit of difference in how much they expressed between the different subpopulations, but all had some expression of the plasminogen receptors, which indicates that they can bind plasminogen and possibly aid in the activation of plasminogen to plasmin. So the next thing that we found was that human T cells actually express TPA as well, and when the T cell receptor is stimulated, this expression jumps up pretty quickly. So 1 hour post-activation, we see a big increase of mRNA expression of TPA, which then drops off pretty rapidly again to close to baseline. So another indication that T cells can bind plasminogen and activate it and use it for something. But what could they use it for? So to actually first test out if they could functionally activate plasminogen, we just had some serum-free cell cultures. The first one is a control, which is a serum-free cell culture without T cells, added plasminogen, and looked at plasmin activity in that cell culture using a fluorescent substrate. So that's background, basically 0. And then we added T cells to the same cultures, and we saw a huge increase in plasmin activity. So even T cells, when they're just resting, can activate plasminogen. And when we activated T cells, we saw a further increase of plasminogen activation. Or if we pre-activated the T cells and then added plasminogen, we saw the same result. So this is what T cells look like when they're actually activated. They move around and they start interacting with each other. So they form these little clusters of T cells. So these T cells were activated with little beads that you can see in there, and these beads have an antibody on them that can stimulate the T cell receptor and the co-receptor. And we see these activation clusters, as we call them. And interestingly, if you add plasmin or plasminogen to these cultures, the size of these clusters increases. So the first bar graph is the control, and then the size of these clusters jumps up when plasmin or plasminogen is added. Which is the first indication that something changes during T cell activation when plasmin or plasminogen is present. Then we looked like, how does this work? And we found that the plasminogen receptor annexinate-2 was actually cleaved by active plasmin, as shown in the last 2 lanes of this Western blot, which has been reported in the literature. When it's cleaved, the receptor complex falls apart and is internalized and can affect signaling inside the cell. And using siRNAs, we were able to knock down to very low levels the annexinate-2 expression and compare it to like a control condition where we have non-targeting siRNAs. So in our control conditions, we have our normal effect. We have plasminogen to the activation cultures, and we see an increase of these clusters. And if we knock down annexinate-2, we completely lose our effects. Still see some clusters, maybe a little smaller than the control condition. So even there it might have some effect, but the plasminogen effect is completely gone. So we're jumping a little bit in the plasminogen system because my PhD supervisor was originally really interested in neuroserpin because he used work on neuronal cells. And neuroserpin is an inhibitor of TPA, which is an activator of plasminogen. So less neuroserpin actually in these experiments means more TPA, more plasminogen activity. And similar to what we saw by just directly adding plasmin, we saw an increase of the cleavage of the annexin 2 receptor, so a very similar effect. And when we knocked these cells down and then activated them, we saw an increase of T-cell proliferation. So what we have so far is that plasmin, when it's present in T-cell activation, it increases the cluster size and T-cell proliferation. And the way we measure this is by loading up these cells with a fluorescent dye that dilutes every time the cells divide. So in this graph, the first peak, the brightest one, is undivided cells, and in blue is our control siRNA. And then in red is our neuroserpin knockdown condition. So, sorry, yeah. And then in the second peak, the cells have divided once, and so on. And if we plot these data, we see that when we compare the neuroserpin knockdown to our control condition, that more cells have divided when neuroserpin is knocked down, and more cells also have divided 2 or 3 times compared to the control condition. Just to quickly summarize this bit of research, so we have neuroserpin that can inhibit TPA, so if you take that away, you have more TPA activity, more conversion of plasminogen to plasmin, the cleavage of the annexin receptor complex, which is then internalized and can affect the actin cytoskeleton, which plays a role in the T cells moving around, forming these activation clusters, and down the line, T cell proliferation. So the next part of the research is focused on the chemokine CCL21, which is very important in trafficking of immune cells to the lymph nodes across high endothelial, directing those cells to the correct zone in the secondary lymphoid organs, and it's also associated with chronic inflammation in non-lymphoid tissue. So CCL21 is similar to another chemokine, CCL19, and they both bind the same receptor, CCR7. But it triggers different intercellular signaling pathways because the affinity for the receptor is different. And also CCL21 is different from CCL19, that it's cell surface bound, while CCL19 is soluble. This is because CCL21 has an additional 40 amino acid C terminal extension that is highly positively charged and can bind to negative charges of extracellular matrix. But when this domain is removed, it's more like CCL19 and it becomes soluble, and also the structure domain changes and increases the affinity for the receptor. And we found that plasmin can cleave CCL21 pretty specifically. So on Western blot you can see a smaller band appearing when CCL21 is incubated with plasmin or plasminogen and TPA. And using mass spec, we actually found the cleavage sites, or the main cleavage sites in CCL21 that removes this positively charged tail of CCL21. So the next thing we did is look at how this affects dendritic cell migration. So we set up an assay where we have a gel, and in that gel we can punch holes, and we can either add a control or a chemokine, in this case CCL21, which we're interested in, and cells, and look at how they respond. And this is what it looks like. So we have 2 conditions, CCL21 only and CCL21 with plasmin. So in the CCL21, based on what I just presented, we expect that the CCL21 sticks to whatever is around. So there won't be much diffusion, so less migration. And when we add plasmin, theoretically, the C-terminal domain is removed and CCL21 can diffuse and attract the cells. So this is what it looks like. This is over a course of 6 hours. So in the first few hours, the gradient is being established, and the cells start exploring and looking around. And then at the 2-hour point, we see a big difference between the 2 different conditions. So when plasmin is present, you can really see that the cells respond really strongly in the directional migration towards where the CCL21 is diffusing from. But what about bound CCL21? Because that has an important role as well. Well, it's involved in the T-cell arrest on endothelial cells, so they can move from the bloodstream into tissues like the lymphoid organs or other tissues where they need to be. And it does so by activating the receptor and then inducing activation of the TG molecule LFA1. So this is work that has been done by James Hucklesby when he was doing his honors degree. And in this instance, we had an endothelial cell line growing in little channels on a plastic plate, cell culture plate, and we can flow over T cells or with and without CCL21. So the CCL21 was flown over first, the endothelial cell, so it can bind to the cell surface, and then the T cells follow after that. And this is what it looks like. Yeah, there we go. There we go. So in the control condition, you see that the T cells bind for a short time and then roll over the endothelial cells and then get swept up again. But if CCL21 is present, they really stick down to the endothelial cells. And we can plot this in a graph so we can easily see what the differences are. So these are presented as the maximum relative to the maximum T-cell adherence that we saw in the assay. So with CCL21, we see an increase of T-cell adherence to these endothelial cells. But if we switch the assay up and have CCL21 in both conditions, but in one we add plasmin, we see that we lose our effect. So plasmin cleaves off CCL21, gets removed by the flow, the capillary flow, and the T cells don't adhere anymore. So far that all fits really nicely with what we were hoping for. Just to summarize this, so T cells can bind plasminogen and potentially enable this to bind it when they're in the circulation and carry it over to different tissue environments. And they can activate it by the release of TPA. The results also point for a role for plasmin in regulating the availability of cell surface CCL21 and soluble CCL21 as well, so 2 different levels. So this suggests that the immune cell actually can use this proteolytic system to use it for a wide-ranging kind of effects. Lastly, I want to share some preliminary data that might spark a little bit of discussion, but I thought it was really interesting. So I work in an immunology lab that's really interested in cancer immunology. And there's been a few papers published, one by Rob himself, that dendritic cells can use plasmin to increase phagocytosis of cells. So we looked at this in the context of melanoma cancer cells. So these melanoma cells I killed using heat and then stained them with a Alexa 488 dye that binds to the amine groups of proteins. So it's a very unspecific binding, binds to any protein that is available basically. And then these were co-cultured with living dendritic cells. So in a control condition, we can see the melanoma cells with Alexa 488. You can see in the background the dendritic cells, and in cyan or in magenta there's the lysosomes, and we can see a small overlap of a weak 488 signal there. But if we pre-treat these cells with plasmin, the signal is very, very bright. So what does it look like when these cells actually endocytose these melanoma cells? Well, they don't actually take up the whole melanoma cell. They seem to attach to them and then nibble off these little bits and transport them through the cell body, and then process them in the lysosomes. And here's the video again. So in purple we have the cytoskeleton, the actin cytoskeleton. Yellow is the nucleus, and in cyan we have the melanoma cells. Which made me think, is there a size-dependent thing? So is it easier for these cells to take up particles of a certain size? And we have an experiment here where I had different size of latex beads, 3 micrometers or 10 micrometers. And there seems to be a difference in how efficiently they take up these beads, because with the 3 micrometer beads, they gather a lot of them and are pretty quickly transport them through the cell body to where we expect the lysosomes are. And with 10 micrometer beads, they attach, but they seem to really struggle in taking up these bigger beads and transport them. We also quantified the amount of Alexa dye they take up that was bound to the melanoma cells. So we have our control condition without plasmin as our baseline. And then when we add plasmin or pre-treat these melanoma cells with plasmin for 30 minutes before washing them and adding them to the dendritic cells, we see a big increase, a threefold increase of fluorescence intensity. If we add a high dose of alpha-2 antiplasmin after the pre-treatment, we don't see any effect. But if we add alpha-2 antiplasmin from the beginning, during the treatment of the melanoma cells with the plasmin, we completely lose the effect of the increased endocytosis. And then lastly, we're looking at some functional effects. Can these cells activate T cells? So we have in this instance, a CD4 T cell clone. So it's a very specific clone that recognizes an antigen of the melanoma cancer cell line. And when we pre-treat this melanoma cancer cell line, we see a big increase in the T cell activator CD134 after 48 hours of co-culture. Now the next steps, because it's still work in progress, is looking in the same assay at CD8 T cells and other aspects of T cell activation like proliferation and functionality of these T cells as well. So that brings me to the end. I just want to acknowledge my old PhD supervisor, Nigel Birch. He really started this research, firstly being interested in neuroserpin and down the line in plasmin. Sadly, he passed away in 2018. But I want to acknowledge Rod Dunbar as well, who is the head of the immunology lab that I'm part of, and my other collaborators, James, Kate, and Scott, and the funders, the university and the Marston Fund.